for coming. And also hello to everybody who is connected remotely. This session that we do today, it's part of the Friday talks at the Marine Science Institute, but it's also part of the symposium, which is a series of marine talks that are organized by different centers of marine research in Spain. Today we have Ramiro Logares as the speaker. Ramiro did the degree in biology in Bariloche in Argentina. Then he moved to Sweden to perform the PhD at Lund University. Did the first postdoc still in Sweden in Uppsala, and then he moved here to the ICM in 2010 when he has had all the different grants that you may have. Finally, he got the position in 2 years ago. When he defines himself, Ramiro says in his biographical sketch that I am leading a research team on microbial computational ecology and evolution. And I think this summarizes very well what he is. He's a microbial ecologist, but he uses a lot of computational power, also with strong interest in big data to analyze patterns in the environment. So, Ramiro, thank you for your talk. The stage is yours.
So thank you very much, Ramon, for the invitation. I was checking the other day, the last Friday talk I gave, it was 2010, so it's been a while. And I think, yeah? There, no? It's better? Okay. Yeah. So it's been a few years already. So, I mean, at that time I was presenting something on lakes, and today it's going to be more about microbes and the ocean. So the ocean is populated by a huge number of microbes, and I don't think you are an audience that I need to convince to about that. But we have 10 to the 30 viruses, 10 to the 29 prokaryotes, 10 to the 28 eukaryotes, and so on. So to give you a reference, I mean, there's about 10 to 24 stars in the universe. So it's an amazing number of microbes. And in terms of biomass, I mean, microbes are the most important component in the ocean as well. There's a recent estimate saying that about 60% of the ocean biomass is microbial. So this is something quite important as well, because this is something we don't see, but they are predominant in terms of biomass as well. Then when we think on ecosystem function, microbes they have a fundamental importance as well. They are responsible for about 50% of the planetary primary production. They are crucial for the marine trophic webs, and then also fundamental for biochemical cycles. Here what we have is chlorophyll in different parts of the ocean, seen from space, and this comes all from microbes, basically. Now, we could think that this was known for quite a long time, but actually the importance of microbes in the ocean is something that became evident not so long ago. So still could be considered something relatively new in terms of knowledge. So let's say that during the past decades, 50, 70 years, we learned a lot on microbes on the Earth, in the ocean as well. And we could answer several questions, but there are still several quite interesting questions that are still unanswered, or we have a partial knowledge about them. So in this talk, I'm going to just address 3 of them. And it's not going to be a comprehensive development of each of these questions, but mostly I'll try to highlight some of the research we've been doing. So the first question is, how do microbes evolve and form populations and species? Second one is, which microbes interact and how? And then the last one is, how much functional redundancy is present in microbial ecosystems? So let's start with the first one. What we see in microbe is that there has been a massive evolutionary diversification. In terms of species, there are estimates of 10 to 12 prokaryotic species on Earth, and 10 to the 12 in the ocean. Remember, that's species, not cells. I mean, it's a huge number. When it comes to genes, there are also estimates of about 45 million for the surface global ocean. Unigenes is unique genes or non-redundant. Then with the same data and changing the bioinformatics pipeline, we could increase this number of genes for the global ocean to 248 million. So the number itself is just to show how many are there. So all these genes represent all the diversification that has been going on. And if we were looking at more detail, we would see more gene swells. And then the question is how this diversity emerged. There are different mechanisms. I'm not going to go through all of them, but I'm going to mention adaptive diversification as one of the most important mechanisms. So adaptive diversification is basically diversion or heterogeneous selection, driving populations apart, and then eventually leading to new species. There are a few examples from macroorganisms, especially birds. I mean, there are quite a few examples, but one of them is from, for example, this bird from the Hawaiian Islands, that from one single ancestor, there were adapted species that emerged. I mean, some species are specializing in, for example, picking nectar, others in insects and crushing seeds and so on. And the size or the shape of the peak is showing, let's say, the different niches that they have. When we move back to microbes, this process of adaptive diversification can be facilitated by their huge population sizes, I mean, the number of individuals within populations, and also the large genetic diversity that is contained within this population. So their reaction to selection can be faster than, for example, in multicellular organisms. So now I'm going to tell you a story, something that we have been working here at the ICM, and it's about the diversification of tiny little ocean predators. So I'm presenting you here MAS4. MAS4 was discovered by Ramon about 20-something years ago, I guess. And it's basically a little cell with a flagella. It's predominant among heterotrophic flagellates. They can be up to 9% of this group. They have small cell size. They are planktonic. They eat bacteria. They are important in the marine food web. They channel carbon energy to upper trophic levels, and they are globally distributed. Now, when we look at the phylogeny of this group, here we have all the stramenopiles, here we have MAS4, and then we have different species. Now, when I came here to the ICM in 2010, we were discussing with Ramon whether these were ecotypes or species. But I'm not going to go further on this. I'm going to just tell you more about this later. To investigate MAS4, we use data from large oceanographic expeditions that most of you know, Tara Oceans and the Malaspina expedition. And the first thing that we saw when looking at the distribution of these species based on metabarcoding, looking at the small fragments of the 18S gene, how they distribute in the ocean, we saw that we could identify the different species, at least 4 of them, not all of them, which is MAS4A, B, C, and E. And what was interesting was that we could start to see different distribution. It wasn't that they were kind of randomly distributed, but they started to show some patterns. As you can see, for example, MAS4A was more present in subtropical areas, then MAS4C seem to be more predominant in tropical areas. So we start to zoom into that. And actually, this is one example where you could see a little bit of a transition between, for example, MAS4C and B in tropical areas to a higher predominance of MAS4A in subtropical areas. So we start to think that they could have different niche temperatures. For example, MAS4C and B, more present in tropical zones, MAS4A, most present in subtropical zones. So then we run some network analysis, co-occurrence and co-exclusion. And this tiny network here shows for example, that MAS4A is co-excluding, as we were expecting, with MAS4C and B, and MAS4C and B were co-occurring. So this was a quite interesting pattern, and then we continue exploring the genome, trying to see where we could find some hints of this distribution there. But for that, I need to tell you something before, which is that these species are very well differentiated. I mean, I mentioned before that we thought that there might have been ecotypes, but not really. When we look at the average amino acid identity between the genomes of these 4 species, the amount of divergence was ranging between 51% to 71%. So this indicates very well-defined lineages. They are not really ecotypes from the same population. Then we run phylogenomics, and we compare with the average amino acid identity, and we got the same, let's say, evolutionary diversification pattern, with MAS4A and B being more closely related than C and E. So these species are genomically quite different. Then we said, well, let's dig in more into the functions that are encoded by this genome, these 4 species. So here again we have MAS4A, B, C, and E, and then here we have the different, let's say, the annotation of the genes from these genomes using broad metabolic categories. And yeah, function and node, interestingly, is the category most abundant, but this is the proteus world, and this happens. But many other categories, we didn't see a big difference. So in general terms, we couldn't see that the genomes of these organisms were substantially different. So then we thought, okay, let's dig in more into genes that might be related to some ecological function, because perhaps there could be differences between these species. So we started to look at the genes in the lysosomes, and the lysosome are organelles, like here, that they contain hydrolytic enzymes. About 60, but depending on the organisms. And the lysosomes are involved in the digestion of biomolecules or other, let's say, biomatter, let's call it. So they are pretty important for phagocytosis and for autophagy. And then we started to think, well, perhaps, there could be, let's say, the enzyme composition of these lysosomes might be modulated in the different species in order to, let's say, specialize or eat different food items. That was one of our hypotheses there. So then I'm focusing here in the glycosidases that were present in the lysosomes, and those are hydrolases that will attack glycosidic bonds, like in carbohydrate, glycoproteins, and glycolipids. And basically, they are quite important in the degradation of food. And remember, these little organisms, they eat bacteria and perhaps other little protists. So what we wanted to do is to see whether different GH or glycosidase repertoire could be linked to different capacities to degrade prey by the different MAS4 species. So again, here we have MAS4A, B, C, and E. We have the number of genes for each of these species. And here we have different carbohydrate active enzymes. What we saw is that there were different numbers of genes for these different MAS4, although the proportions were not that different between them. But the numbers, let's say the total amount of genes, they were differing. Now, one interesting thing was that when we were grouping the MAS4 based on the repertoire of glycoside hydrolases, the grouping was not reflecting the phylogenetic history. Let's say we were having MAS4A and MAS4C grouping together based on the repertoire of GH, but those are not the most closely related organisms. MAS4A and C are not, this is a phylogeny, so these are not the most closely related. And the same thing with MAS4B and C, they were grouping based on the GH composition, but they are not the most phylogenetically related organisms. Then we start to link this with what we saw before of the geographic distribution. And one thing was quite interesting was that species with similar GH repertoire, A and C, here we have them, they were co-excluding each other in the ocean. While the species with similar GH repertoire, C and B, they were co-occurring. And here we have the previous network that well, we did based on geographic distribution. So using this information, we try to put forward one hypothesis that is trying to explain how MAS4 diverge, at least this species. So we think that there was an adaptation to different temperatures, and also that the competition between species have promoted the, let's say, the biogeographic patterns that we see now in MAS4, as well as their species diversification. And remember, this is one hypothesis. There could be other ones, but this is one of the most parsimonious ones. So we have the last common ancestor of MAS4, and then from that last common ancestor, we think that MAS4E was the first that diverged, and that species adapted to or remained in cold waters. We don't know whether it was a cold or warm water species at that time when this happens. Then here we have the last common ancestor of A, B, and C, and we assume it's a warm species, probably perhaps living in the tropics. Then from this last common ancestor, species C diverges and stays in warm waters because it might have been a better competitor there. I mean, it's adapted to that particular niche. And then here we have another diversification point. To avoid competition with C, I mean, these other two species, they diverge in their niche. Species A adapts to colder subtropical waters, while species B stays in the same geographical area that is populated by C, but it might have changed its niche by diet modification. By specializing in a different type of food, then it could coexist with C. So yeah, this is our hypothesis. There are other ones, but it seems that it's one possibility. Now, I've been talking about species so far. So MAS4A, B, C, and E, they're well differentiated groups. But what about populations? I mean, populations are all the different individuals that are present within each of those species. And why this is important? I mean, population variation might provide insights on their reaction to natural selection. I mean, if the whole population is clonal, if there is a change in the environment and the potential to adapt to the new condition, it's much more limited, as if the population has a larger amount of diversity. And this is quite relevant in the context of global change, because we don't know how microbial populations will react to changing ocean or changing environmental conditions. So to investigate microbial populations, what we use is the analysis of single nucleotide polymorphisms, or SNPs. Here we have just 3 hypothetical species, and then what we look is at basically mutations between them, so point mutations. We use also something called an index, the FST index, of fixation index. And to explain it is, well, it's relatively quite simple. It's looking at the variation between different populations against the variation within population, and then a division there. But we can have 3 different scenarios. For example, we can have, if this is one species, we can have 2 populations. And when the FST is equal to 1, it means that there are 2 populations that are genetically completely different. Then if we have an FST of 0, it means that those 2 populations that we thought were different are not, are probably genetically are the same. And then, of course, we have a lot of values in between. That's real life. I mean, all those values that are in between is what we get. And in those cases, we see that there's some differentiation, genetic differentiation between different populations. Now, we try to detect SNPs from metagenomes, and I mean, most of you are familiar with metagenomes, but just to have everyone on the same page, metagenomes are try to, let's say, represent most or a good fraction of the genomic information from a microbial community. It's not everything, but it's representative from the most abundant organism. And basically what you have is millions of little sequences from different genomes in your metagenome. So we have different metagenomes that we're taking from Tara Ocean. And what we do is to compare or map those metagenomes against one reference genome. And in this case, this is MAS4. So we compare the different metagenomes against one genome, and we detect point mutations. And in this particular case, we use MAS4, and we have 82 metagenomes from Tara Ocean. The interesting thing was that when we started to do this analysis, we weren't very sure what we were going to find. Perhaps everything is randomly distributed, maybe not. But we started to see some structure. Here we have the different MAS4 species, and here we have the distribution of the FST values that we recover. Now, when we have FST of above 0.15, we could say that there is a high differentiation. If it's between 0.15, 0.25, this is a high differentiation between populations. Above 0.25, it's very high. So serum values are above 0.15 for MAS4A, B, and C. So this was a hint that it seems that there is population structure here. So we need to dig a little bit more. There was not so much variation for MAS4E, but I will tell you later a little bit more about that. I mean, it's a species of cold water, so it wasn't well represented in our data set. Now I'm going to talk a little bit of MAS4A. I mean, we did for the other species as well, but I mean, with one, I think it's good to convey the message to you. So we try to identify populations in MAS4A. This is the map showing the sea surface temperature. And each of the dots is one station from Tara Ocean, from where we took metagenomes. The color of each station is related to one population that we define. So here what we have is the FST distances that were clustered in a hierarchical clustering, and then the abundances of the different, of MAS4A in the different samples, and then the number of SNPs or mutations that we could identify in different samples. So what we did is to define groups, and those groups represent potential populations. I mean, everything with an FST below 0.15 was defined as a population. So we could define one group that is the blue, let's say the blue group, or the blue population, which is here, which is the most abundant, and it represents let's say, the actual distribution of MAS4A, what we saw with metabarcoding. So this was making sense. This abundant group represented, let's say, the most abundant MAS4 species. Let's call it like that. But then we found another population in the Mediterranean Sea. Sorry, I mean, this was cut from the slide, so sorry about that. The Mediterranean Sea seems to harbor a population of MAS4A that is quite differentiated from the, let's say, global ocean population that is present in the subtropical areas. And then we have another population that we are not sure if it's a population or not yet. We are still discussing. I mean, the red group, which is there, it's in areas where, if you remember before, I mean, this is the tropical areas is where MAS4C is dominating, and it's MAS4B there as well. So we don't know whether this represents a population with smaller abundance that is present in tropical areas, or it might be an artifact of when we do the mapping of the metagenomes that we are getting signal from another MAS4. So we are digging on that, but at least two populations seem to be well differentiated for this species. And some similar examples are present also for the other MAS4, but I'm not including them here. Then we were looking at let's say, the genes of the different MAS4 species, trying to see where we could see any signature of selection. So for that, we normally use this, let's say, the ratio between non-synonymous and synonymous substitutions for different genes. And in this case, when we have, let's say, a ratio above 1, it's a potential indicator of positive selection, adaptive evolution. So here we have, again, the 4 species, mass for A, B, C, and E, and each dot here is a gene. And then what we were doing is putting the counts of non-synonymous versus synonymous substitutions for each of those genes across the 80-something samples of Terra oceans. What we start to see is that there are some genes that where the number of non-synonymous substitutions is higher than the number of synonymous substitutions. So perhaps those genes are or could be under positive selection. Especially in mass 4, there are a few less in the others. So we are digging into that, and we're trying to see whether those genes could explain some of the population differentiation we see. And these are good candidates to try to understand the adaptive diversification in these populations. So if you, well, I mean, most of that work was done by Fran Latorre, who's finishing his PhD with me. We've been collaborating a lot with Ramon, with Olivier Jaillon, and then part of his work was recently published. You can have a look if you like. And the second part, let's say the population part, is still ongoing. So hopefully in some month there will be more news. Now, one thing to think about is when this variation emerged, because I have been talking about lineages, populations, so these are different temporal scales. When we talk about phylogenetic groups, defined phylogenetic groups, we're mostly talking about microevolutionary patterns, things that happened some time ago. It's difficult to put a number, but it's not something that recent. Diversification of different mass 4 could be several 100,000 years, million years, I don't know. I can't give a good number, but it's not something that is occurring right now. But then when we are looking at populations, we are looking at microevolution. These are processes that are occurring at smaller time scales. And this is interesting because at smaller time scale, ecology and evolution might be interacting. So evolutionary change might affect ecology, ecology might be affecting evolution, and that's something that is, we think is quite interesting. And this can occur at contemporary time scale. So evolution might be a process or is a process that occurs at contemporary time scale. Contemporary means, let's say, decades, we could say, or years. And, well, one good example of that is actually with COVID-19. So here we have, let's say, the diversification of COVID-19. And I'll put you a small video. Let's see if it works. So COVID-19 started about 2 years ago there, and there was a little diversification, but very rapidly, new strains started to pop up. You can see that each of these points that pop up are new strains with different nucleotide composition. We can see Delta showing up there. We can see, I think, Omicron is already appearing there before all the diversification of Omicron. Then we have Delta, huge diversification going on and dominating the population. And then we can see Omicron there is still not very common, but then it starts to increase. So this happened in 2 years. I mean, to me, it was amazing to see that we could actually track evolution in almost real time. Of course, this is a virus. I mean, we cannot translate this to a proteus, to a bacteria, because, I mean, the variation is slower there. But it might be possible that the amount of contemporary evolution that we have, for example, in bacteria in the global ocean, it's more important than what is currently acknowledged. And this is something we started to look at in a couple of projects. So the question is contemporary evolution going on? We can detect it in marine microbes. And these results are quite preliminary, and this was done mostly by Patrick, that is doing the master here with me. So what we did is we could retrieve a SAR11 genome from Blanes Bay using long-read sequencing, PacBio. And then we said, well, let's try to see where we can see the change over time in that genome. And for that, we have 12 years of metagenome from Blanes Bay. So we said, let's compare this genome during 12 years. The genome was well complete, and then what we use is, again, this ratio, the NDS that I mentioned before. We compared non-synonymous versus synonymous mutations. Here we have the ratio, here we have the year from 2010, 2009 to 2020, I think. Different seasons, and we have one hypothetical protein that we put here as example. So what we can see is that there's a big variation on the NDS ratios over time. I mean, for that specific protein that belongs to SAR11 in the different seasons, there's variation in the ratios, but we could see that in some cases, it seems that there is a trend over time. So if there is an increase of the NDS over time, this could be a suggestion of adaptive evolution. But again, I mean, we are double checking this, we are working on that, so we need to confirm that. But it would be very interesting to see if there is an increase in this ratio over time that could be indicating adaptation, at least in one specific gene for one genome, to changing conditions, potentially. So this led to other questions, or it's leading to other questions. So for example, if we assume that we detect evolution at Blanes Bay, which is one spot here, what does this represent? Is this evolution in this single site? Is the whole Mediterranean Sea? It's a bigger area, so we don't know yet. But we're trying to see where we could identify the extent of the process. Then we're interested to know what are the exact phenotypic effects of those evolving genes, and also how microbial population will evolve as response to global change. And this is something we are working with Georgina Brennan, who is a postdoc here at the ACM, working with me in a couple of projects. And we run an experiment in Sweden because here we had COVID, so it was complicated. So during 14 months, we tried to expose communities, microbial communities, to different conditions. And we are now in the process to see where we can detect genetic changes in some of them that could be indicated adaptations to the different treatments. I mean, treatments simulating global change, increased temperature, for example. So I cannot tell you more now because we are analyzing data, but hopefully, I don't know, in a few months, there will be more results here. So we move now to the second question about which microbes interact and how. And this is a general depiction of, let's say, trophic interactions in the ocean. We have phytoplankton, so plankton fish. I mean, this is a typical, let's say, classic transfer of matter and energy in the ocean. We have particulate organic matter, sinking, and then we have some matter that is used by, for example, bacteria that are then eaten by protists, and this is channeled again to the, let's say, to the main trophic web in the microbial loop. The interesting thing here is that there are a lot of interactions here going on between microbes. So these little guys are quite important to maintain the interactions or the trophic interactions between larger organisms in the ocean. But the thing is that we know very little about these interactions. And this is mostly a black box. We know a few, but most of them are unknown. And this is something we've been wondering a few years ago, and we wanted to quantify how much we knew. Let's say we know that we don't know a lot, but how much we knew. And what we did is to, let's say, do a bibliographic search. So there were 2 students working for 1 year, looking for papers. I mean, it's a lot of work, going through about 500 publications and registering the interactions. And this paper were from the last 150 years. And after all that work, we came out with this network. I want to just stress that this was mostly focusing in protists. So protist-bacteria interaction, not bacteria-bacteria. So this network came out, and this network contains all the associations or, let's say, interactions that we could detect. Each of these nodes is a group, and then we see that taxonomic group. Some groups like dinoflagellates, ciliates, diatoms, they are more represented because they have been studied most. But then we have all these other groups that, they are barely known about what they are, with whom they are interacting. And most likely they are interacting with several other microbes. So to try to address this lack of knowledge, let's say, on microbial interactions, what we do is to use association networks. And association networks can tell us good hypotheses of interactions. Of course, this is not, they do not prove that the interaction is ongoing, but they can tell us if potentially there might be an interaction between two microbes. So basically what we do is just look at correlation between microbial abundances. These are two simple examples, two microbes which are correlated over time in their abundance, or two microbes that are not that well correlating or anti-correlating in abundance over time. And with this data and some fancy methods, computational methods, we end up with things like that, which are the networks, where each dot or node is a species, and each connection represents a potential interaction, positive or negative, co-occurrence, co-exclusion. So now I'm going to present some results of what we did in Blanes Bay as well. We use data from 10 years, and we use that to build networks. We look at prokaryotes and protists, picoplankton, nanoplankton. We use metabarcode in there, and we focus only on the strongest associations. So I'm jumping directly to the result there. Since we use only the strongest associations, we basically build a core network, let's say the most important, or what we think could be the most important interactions there. We have about 259 species, 1,400 associations, and then even though those 259 species were 9% of the richness, they were more or less 50% of the abundance. And most species that we found were seasonal. Here we have the networks. Again, each of these points or dots is a species. The color is the season, winter, spring, summer. And you can see that the winter group seems to be better connected than the others. So it seems clear that, winter associations seem more prevalent than the associations in the other seasons. Now we translate the same network. This is a different plot of the same network. This is to just show the different species that are involved. We saw bacteria were the most prevalent in that network, and that protists were less, about 30%. And bacteria and protists, they tended to be associated with bacteria. So this shows more or less the distribution of associations we found. And of course, all these are, I would say, good hypotheses, but they need to be tested and confirmed. We also saw modules, and modules are a group of species, or OTUs. I didn't mention what is OTU, Operational Taxonomic Unit, a proxy of species. So OTUs are or species that were more connected to each other than to the rest of the network. So they represent tight groups. Here we have different modules, module 1, module 2, 3. The color, again, is related to the different seasons. For example, there are several winter modules. There are one summer module and one autumn module as well. So these could be, these are good candidates to search for quite strong associations within this network. The idea is to investigate at least a few of these associations. But then I've been showing you static network, just snapshots. This network is more or less the same or similar to what I presented before. It's a static network, 10 years of data, and you get that snapshot. But actually, you could open this up and get a specific network for a specific month. And when we do that, we start to see that this single object starts to show different patterns. For example, this orange cluster, which is present in warmer waters and disappear in colder waters. So networks are changing over time. And this is a kind of video that Ina, my former student, did, trying to show how the different clusters, the blue and the orange, they appear and disappear over time. So that's something we need probably to incorporate in our knowledge now, the changes in the associations or interactions that occur over time. Then, I mean, this is, well, continuing with network dynamics. I mean, here we have, in this panel, we have the number of nodes for the networks for each month during 10 years. And we see that the number of nodes in the network, number of species in the network is changing. And this is the number of connections or edges over time as well, the same, different years. Each point here is 1 month. And we see that the amount of edges well, changes over time. There are more edges in winter, less edges in summer. So this points to a change in, or a dynamic, quite dynamic network. And these are other indices that are used in networks like edge density, transitivity, average path length, and so on, that they also show seasonal behavior. So the interesting thing is that over 10 years, the network is showing recurrent patterns. It's not that every season you get a new network showing up. I mean, some things are new potentially, but there are some connections that are repeatable, especially in winter, where there are some associations which are recurrent. And the cool thing, I think, is that this network that we see, or for example, in one specific season, most likely will disappear until next season, and then will sort of re-emerge and form again. So networks also change across space. This is just to give you a glimpse of what we did here with, actually there's a tree there that you don't see. It's about 400 samples and 400 networks. This is data from the Malaspina expedition and also from the hot mix expedition in the Mediterranean Sea. And these are different depths of the ocean. I mean, samples for surface, well, DCM, mesopelagic, bathypelagic, and so on. So based on that, we built networks. And what we did here is to build networks for specific basins or sub-basins, like, let's say Mediterranean Sea, and basins like North Atlantic, South Pacific, and so on, and for different depth layers. Epipelagic, mesopelagic, bathypelagic. Now, we won't go in detail on this because we could spend half an hour looking at this, I think. But what we need to look at is basically the colors here represent different taxonomic groups. And then what we can see is that if we move vertically in one, for example, Mediterranean Sea, we see a change in the predominance of some groups with depth. And the same with other basins. And when, I mean, this red color are mostly archaea, which is expected, let's say. So we see a vertical change, which is not the same in different basins. And we see, let's say, a change also in the horizontal dimension of the ocean as well. So networks change over these two ocean dimensions. Well, I want to thank Ina, who did a good part of this work. She's now a postdoc in Paris, and my former postdocs, Anders and Marit, that did also a good part of the work. And now we enter in the last question, which is how much functional redundancy is present in microbial ecosystems. And you might wonder why this is interesting. Well, I mean, there was a lot of discussion during the last years of whether microbial communities in the oceans are redundant in terms of function or not, but I will tell you a little bit more in the next slides. So functional redundancy. The idea is that we can make an analogy between a team and an ecosystem. A team and an ecosystem, they both need to work, right? And the question is whether the species or the players in the team, they are essential, they are redundant or not. I mean, what happens if we change the players of one team, do we get the same results or not? What happens if we change the species in one ecosystem? Do we get different functions or not? So here is an example with Barça and different formation of Barça over time. The question if Barça functions or not is not out of the scope of the talk.
How this week?
Yeah.
This week?
So here you have different players in the last few, let's say, seasons. So different formations, and if you start to look at them, I mean, you start to spot some players that are present in all of them, like Frenkie de Jong and Jordi Alba. They appear in most of them. Then we had one that we knew that was very important, but it's gone. We have it here. And then there are other players that I know it sounds bad to say they are redundant, but they could be changed. And well, the question is whether they generate different results or not. But this is the idea. What's the effect of changing components on the function of a system? A team could be seen as a system, and an ecosystem is a system. So in order to look at this on microbes, what we normally do is to compare taxonomic similarity with functional similarity. So let's say we have a bunch of different communities, and we try to see whether the change in the taxonomic composition of the different communities leads to a change or not in the function of that community. And how you measure function, well, we will talk about this in a bit. Now, if you see that communities change in terms of taxonomy, you have different taxonomic change between different communities, but functionally the communities don't change, we say they are quite redundant. I mean, there are different components that can perform the same function. But if we see a relationship between taxonomic similarity or taxonomic change, let's say, between communities and functional similarity, then communities are, they have a lower redundancy. So we were investigating a little bit of this on two neighboring time series, the one that I mentioned before, Blanes, and the Vanille-sur-Mer time series. We use 7 years of synchronized monthly metagenomes. We look at surface waters, and we analysis of synchronic dynamics. And this is the question: how we define function? Yeah, there was a huge or large debate on what's the relationship between, let's say, taxonomy and the function of communities. I'm discussing a little bit with Pierre Galland that he's in Banyuls, and he's been working quite a bit on that. We were talking about on the level of resolution that we want to look at when we define function. So let's say we could define function of a microbial community in terms of, let's say, in terms of all the metagenomic content that we have from that community, in terms of all the predicted genes, and a bit of a spoiler, what comes here is all metabolic functions. This is more fine-grained, this is more coarse-grained. Here again, we have taxonomy, and in the y-axis here, we have different, let's say, proxies of function. Now, when we look at the relationship between taxonomic differentiation of similarity, as you prefer, with all the metagenomic content, we get this very good relationship above the 1-1 line. I mean, this is a very good, let's say, association there, or correlation. Now, when we look at all the predicted genes, that means, I mean, we are not looking at the whole metagenomic content. We're just using the genes that we could predict. Then we get the same relationship. Now, when we look at all the metabolic functions, things change. So these are broad metabolic functions that were annotated with this specific database. And as you see, I mean, we have this relationship above the 1-1 line, and then we went below the 1-1 line here. And again, taxonomic differentiation, I mean, substantial or larger change in taxonomic differentiation is leading to a smaller effect on the functional differentiation in terms of this broad metabolic function. So defining the level at what we define function is important to interpret the results. So I'm continuing with this now. let's assume that we have comparable communities, microbial communities, in these two time series, and that they experience similar environmental conditions. So we have these two hypotheses. I mean, if we have a high redundancy scenario, we could have a low synchrony in taxonomic turnover in these two sites, but a higher or moderate synchrony in functional turnover defined as broader taxonomic functions. So we are in this coarse-grained scenario here. Then we have this other hypothesis, which is a low redundancy, where we have a high moderate synchrony in the taxonomic turnover of these two sites, and also we have a high moderate synchrony in the functional turnover. When I'm talking about synchrony here, I'm talking about this. I mean, this is one example with the near K gene, or function, let's call it. We have the two sites, blanes in reddish and black sola time series. And we see that over time, this is time here, we have a good correlation in the two sites. But then we have the AMT gene that is not that well synchronized. One thing to keep in mind is that these two systems are connected. I mean, they are not isolated entities, but they are connected through currents. This is a nice plot that Sergio prepared from the Cosmo site here at the ICM. And when looking at different degrees of synchrony, we started to see, well, let's look at the synchrony of species, genes, functions, but let's start with species. We have, I mean, in the two time series, we have about 11,000 species or OTUs. And then we could identify different levels of synchrony. There were a few species, 1%, that were highly synchronous between the two sites. Then there were about 17% of species with medium synchrony, low synchrony, that can be identified here. It's most of the species, actually, that have low synchrony, and a few of them as well with negative synchrony. This is for species, and then when we look at genes, we get something kind of comparable. So what we observe with species, we see it also at the gene level. I mean, the amount of synchronous genes is about 3%, 25 are, they have a medium synchrony, and so on. And moving to functions, again, it's a kind of comparable distribution. Here we have an average of 45,000 functions from different databases like EGNOC, PFAM, COG, and so on. These are different databases that we use to annotate genes and assign functions. So again, the highly synchronic functions was the group with let's say, the lowest abundance, and there were other functions with medium synchrony, which were higher, or more functions with medium synchrony, and most functions, they have low synchrony. So there is a kind of connection between what we see in species and functions. So it looks like we are in this low redundancy scenario, although we're still discussing this, but it looks like the important thing here is that we have different behaviors in the two time series. We have things that are highly synchronic, things which are antisynchronic or with low synchrony. So all those behaviors can be observed there. And then we look at this, well, a number of key marker metabolic functions. We selected 45 functions, and these belong to different metabolic pathways, like, for example, carbon metabolism, nitrogen metabolism, phosphorus metabolism, and so on. And we also see a distribution there. There's a distribution of different degrees of synchrony for different functions. Some functions are highly synchronic. Here is the synchrony degree going up, which is AMO A, AMO C, and B, and near K. And then there are other functions which have a lower synchrony. And this is the taxonomy composition of the genes that contribute to that function. So for each of those functions, there are a lot of different genes coming from different species that are contributing to that function. So we see, for example, this highly synchronous function is dominated by archaea here. So perhaps it's a kind of trivial scenario because, I mean, let's say that the dominance of, or the synchrony of one species is leading to the synchrony of a function. So perhaps more interesting are cases where we have, for example, here or there, where there are more genes from different species leading to the synchrony of function. Although normally we have one or a few species leading the synchrony of the function. Then another thing we've been wondering is whether we could have scenarios where the synchrony of a function couldn't be explained just by the synchrony of the genes that are contained in the function. And this would be something like an emergent behavior, we could say, that the sum of the parts cannot explain the whole, so to say. This could be in a scenario where we could think that, well, I mean, there could be some sort of randomness in the behavior of genes, but then the whole function will show, let's say, a specific pattern because, I mean, that function is needed for a system to function. So what we have here is, I mean, the same 45 functions, each of them. The size of the bubble here is related to the number of genes that is contained or which are contribute to those functions. And we have the gene synchrony. This is the mean synchrony of all the genes contributing to each of those functions. And here what we have is the whole function synchrony. Let's say when here we take into account the whole abundance of the function and we look at the synchrony, while here we look at the synchrony of each of the specific genes that contribute to a function and they take the mean. Now what we see are cases like this where the functional synchrony is higher than, or slightly higher than the gene synchrony. So we might be spotting there some synchronic kind of behaviors in the function that cannot be just explained by the average or the synchrony of the genes in that function. And then we have other interesting examples like this one where the synchrony of the genes contained in the function is, let's say, it's a number that is not trivial, about 0.20 something, but then the functional synchrony is pretty low, or almost below zero. Now here what we are seeing is, or we were looking with Sergio the other day, and then it seems that there is different synchronies going on for the genes within that function that sort of cancel out. And then when you look at just at the whole abundance, then the function is not synchronous, but genes in there, they are. Okay, so I want to, thank, I mean, a lot of that work was done by Sergio, a lot of bioinformatics by Lydia, and then we are collaborating a lot with Pierre, Jose Gonzalez, and also Pep on this work. And probably there will be some new results soon. So I'm having a few take-home messages. So the first is that, most of you are aware of that there's a large diversity of micro niches in the ocean, and those probably are driving population structure. And looking at population is probably something that will become very important, I think, in marine macroecology and microecology in the future. I mean, because now we can do it. Before it was pretty difficult to access up to population variation, but now we can access there. And probably we will get some surprises. Then contemporary evolution going on at time scales that are relevant for us, let's say. It's a bit anthropocentric, but yeah, that's our definition, let's say. It could be more important than we think. I mean, we could have evolutionary processes going on in decades in the ocean, and understanding those will be important for our understanding of the reaction of the ocean microbiome and the Earth microbiome to global change. Then on microbial networks, we need to probably introduce the dynamic nature they have, because they change over time. So we need to start acknowledging that and see that as a dynamic system rather than one static object. About functional redundancy, well, it seems to be quite dependent on the resolution level that we're looking at. So it's important to consider that because otherwise it can lead us to different interpretations. So defining what is a function and how we measure it, it's quite important to understand the system that we're investigating. And then within microbiomes, we probably can have different levels of synchrony. I mean, not just a few behaviors, but probably there's a range of behaviors in terms of synchrony, and that probably could be linked to the importance of specific genes or specific organisms in the functioning of one ecosystem. So I want to thank a lot of people, people at my lab, and then the College of Marine Microbes here at the ICM. Of course, co-authors, because without them it would be impossible to do all this. The Institute, Blanes Bay, the large expeditions, Malespina and Tara Ocean, some institutes that help us with part of the work. Well, our computer cluster that has been working intensively to get those results. And also the funding agencies, and I want to thank you for being here. Thanks a lot.
Thank you, Ramiro, for this very comprehensive and extensive show of your data and analysis. It was extremely interesting. We have time for a few questions, and I will pass the micro around. Celia, she's very fast, as always.
Thank you, very, very nice talk and a lot of work. I was wondering, because when you do this global analysis with Malespina data, and then you rate this distribution basically with temperature, but you have there areas where there is a strong seasonality and others that don't have seasonality. And then you go to Blanes, which is an example of temperate climatology with a lot of with a marked seasonality, then you see difference. I'm wondering how much difference are due in this global analysis, just because in some of them are influenced by seasonality and you sample just once, and then it can be winter or summer, or I don't know if.
So we look at the general. Let's say we have the spatial and then the temporal dynamics for populations. So I was showing the dynamics for mass 4A in the global ocean. And there, of course, there are different seasons, but we didn't take into account the temporal variability there. It was mostly spatial. So at that resolution, we could see that we could identify, for example, different populations. Clearly, one for the Mediterranean Sea, another one for, let's say, the subtropical area of the ocean. And I didn't show it here, but Fran did a similar analysis for the Blanes time series, and we didn't see patterns that were so clear, let's say, for the mass 4, right? There's variation, but we cannot see clearly different populations there. While let's say at the global scale, we could see clearly defined populations. And probably it's because, the reason is that the extent of the sampling, it's much broader, right? It's more likely to find populations, I think, in the global ocean that just in one specific site, although it could happen that, with bacteria, we see that there are populations replacing over time in Blanes, for example. So I'm not sure if I answer, but...
Hey, great talk. I have a question on the first chapter, but the idea percolates through everything. In the first chapter, you find differences in the genes. of the different species of, or you suggest that there are differences in the genes, that this is the evolution, but the linkage between these differences in the genes to the real ecological behavior of the organisms, this is something that is still we cannot get. And this is something that starts to worry me, that we are finding, you may find evolution in the genes, but then how do you explain that ecologically? I mean, what changed in the environment that allowed this evolution? What are they are better adapted to what? This is the...
Yeah, we have the same question. So we're trying, to look at the type of changes, and we can, let's say, assign some different type of changes in a specific protein. So, for example, we could identify some genes that they seem to be more affected by evolution than other genes. So we try to look at them, and they seem to have more non-synonymous changes than synonymous changes. So that can suggest evolutionary adaptation. And then we try to, the next step is to try to see what effects they might have on the protein structure. And this is, we were interviewing with Francesco about this to try to model different type of variants of a gene and see where we could identify some, for example, structural changes. But then, let's say the next step is the experiment, perhaps. And of course, it goes like, quite a bit into the future. But I will say that here we are looking at the first steps to try to address the population variation. And then, for example, with experiments, with modeling of proteins, we could try to understand what those changes mean for ecosystem and functioning. But we could, for example, think that we could have some changes that lead to locally adapted genes or proteins, to function at different temperatures, under different conditions. But,
Hi, thank you. Thank you for your talk. It was very interesting, all the 3 chapters. And my question is for the second. And I am curious to know, what do you include in these interactions that when you did all this search in the literature, what do you include in that word, the several interactions? And if it would be possible to separate them and do individual networks of predation or symbiosis or whatever, if it would make sense.
So the networks they help us to try to identify potential interactions, let's say. So these are all hypotheses of interactions, and of course we can filter out a lot and keep only those associations that could, potentially tell us that, this is a real interaction. But of course it needs validation afterwards, and that's something we didn't do. And probably, will take quite a bit of work to validate in the lab, the interaction. But based on the taxonomy of the organisms that interact, we could provide a hypothesis of the type of interaction. For example, predation could be, or symbiosis. And of course, once we have, let's say, put the interactions into categories, the next step is to try to validate those in the lab. But yeah, we can, based on the taxonomy, we can provide a hypothesis of the type of interaction that is happening. I forgot your second question, I think. Sorry.
It was only one.
I was only one? Okay. Yeah.
I have also a question. I think the comparison between the 2 sites, between Blanes and Banyuls, that's quite interesting. And you started with a map of the circulation of the ocean that the sites are connected. So one would expect much more synchrony between the sites. And still what you show is that the synchrony is extremely slow. No? The majority is low, where I don't know what it means. But basically there is very little synchrony between the 2 sites.
Yeah, it's a distribution. There's a distribution of things, let's say, or synchronies. So a few things which are quite synchronic, and then a lot of things which are not. So the sites are connected, but they are different. And there are some slides that I didn't show, but if you compare the whole, let's say the whole metagenomic content of the two sites, we see that there are similarities, but there are differences as well. So they are not, they are far from identical. They are not two completely different things. And some species, they seem to be behaving the same in the two sites, and some genes, which are, I think it's quite interesting that some genes which are, or functions that are, let's say, based on several different organisms, they are quite synchronized as well. But then there are several others that they are not. And this, one could think, there could be a link with redundancy there. I mean, they are not redundant because they are not synchronized because, I mean, other organs are carrying out that function. Or there could be other explanations. But
if anybody has a last urgent question, and if not, I think we can just thank you again, Ramiro, for your talk
thanks to you
and your
for
presentation.
being here.